Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, our panel of experts will discuss international perspectives on model-informed precision dosing from the data to the patients. Amparo de la Peña, our Vice President of Pharmacometrics Services, and Ana Ruiz, Head of Pharmacometrics at Gilead Sciences, will serve as our moderators today. We want to thank the Pharmacometrics Committee of the International Association of Therapeutic Drug Monitoring and Clinical Toxicology for their official endorsement of this webinar. A few housekeeping items before we begin. We take your privacy rights seriously by registering for and attending this event or participating in the Q&A session. You are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all the questions in the Q&A after the presentation. And then before we begin, I'd like to conduct a quick poll with 2 questions. And I'll give everybody a couple seconds to answer both of those. Okay, I do see a couple of answers coming in. Okay, and I'll end it there. Thank you everyone for your responses. And now I'll hand it over to our moderators, Amparo and Anna, to introduce our speakers.
Thank you, Jasmine. It is my pleasure to introduce our previous speaker, but before that, I wanted to give thanks again to the International Association of Therapeutic Drug Monitoring and Clinical Toxicology for endorsing this webinar. Our first speaker is Dr. Ron Keizer, and he is a pharmacometrician, pharmacist, and software engineer with a passion for developing tools to improve drug dosing in patients. He has held various academic positions and authored several commercial and open source softwares in the pharmacometrics field. He's the co-founder and chief scientist of InsightRx, a San Francisco-based company developing precision dosing software for providers and for pharma companies. So without further ado, I'll stop sharing. And Ron, feel free.
Ron, I think you're on mute, and your slides. Can you try presentation mode?
I'm sorry about that.
Okay, I can hear you now. Cool.
Yeah. Here we go. Do you see my slides now?
Yes.
Yes. Okay, sorry about that. False start. Thanks for the introduction, Amparo. Very nice to be invited to speak here. Yeah, so I'm going to speak about continuous learning in model-informed precision dosing, because I think it's a very important topic. It's also a topic that we do a lot of research within the company, and I'll explain why. So why do we need to learn from our data, and yeah, and I'll explain later how we should or how we could do that. So one reason is that for some drugs we just don't have good models available. Of course, for newer drugs where we might have one model, or maybe not even one model, or we might have a few models, but they're not built on a lot of patients, so they might not be good for the population that you want to use it in. So that could happen. It could happen, for example, for vancomycin, that you actually do have a lot of models, but you're actually missing a model for a specific subgroup or a specific formulation. That could also happen. So then you might want to use the model that's available, or one of the models, but maybe retrain it on that specific population. Another reason, this is also something that we see a lot, is that within or between hospitals or between regions, we actually see differences in PK, and that could also, if you have a specific model for specific hospitals or for specific regions, that could also help improve the predictions. And lastly, I think this is also very important. When you have models and you use them in sort of a flow where you actually update the model based on the TDM that you collect, I mean, it's still important that that model is good, but it's less important because you actually adapt the model to the patient data that you collect. So it's less important over time. But what we want to do is also use the models specifically for the first dose. And for that, you have to have a model that's really unbiased and precise for the population that you want to use it in. So there it's even more important to have a good model. So with that, how would this work in practice? So about half of the hospitals that we work with have an integration with our platform, where the hospital data from the electronic hospital records flows into our platform, and then we were able to provide an advice based on the model and the specific data for this patient. Besides that, that data can then also, of course, be used for learning to improve the model. What's also important is cleaning of the data, of course. Once the data is used to retrain a model, you need the data to be clean, otherwise the model will be biased. That's pretty obvious. Of course, what you also need to think about when you do continuous learning is a way to make the models available again. Of course, you could do it manually, but then it's hard to do it every week or every day even. Maybe you do it once a year or something. But ideally, you want to have this in a more automated workflow. And the last thing to point out here is that you also have to monitor the performance of the system. When we think of ways to do continuous learning, at least how I think of it, and there are more ways to do continuous learning, but this is sort of what we work on in the company. It's basically two approaches. One is based on model selection or model averaging, and the other is really based on retraining a model. So if you zoom in on the model selection approach, you can also split that up again. One is just looking at, okay, in retrospective data, let's say we have 5 models for a specific drug. Which of these models is retrospectively the best model in terms of bias and precision? And that's something that we've done a lot in the past. Also, one of the first features in our platform was based on that, which was sort of an auto select feature where based on the BMI of the patient, so a BMI higher than 40, you would get a different model recommended than a BMI lower than 40. And the same for example, for a renal function, we have specific models for lower renal function versus higher renal function. So that's what we've done in the past. What we're working on right now is more extensive ways of doing that, and one of them is what we call the matrix auto select, where instead of just doing like a really simple splits, you actually make it more of a matrix where you look into certain age groups versus BMI groups, and perhaps versus renal function groups even, which model is actually performing well. And then you see for each model, you see really differences occur where they perform well in some area, but not so much in other areas. And to do this analysis, of course, you need a lot of data to have enough patients in these boxes. But with the data set that we have for vancomycin, this was feasible. And then if you make this into sort of an algorithm, you can then just for every cell you basically select the best model, and that's highlighted here in the colors. So each color is a different model. So we can, for example, see that the Colin model really performs well in the younger age, and let's say the lower weight the patients, while we have a specific model for obese patients that also performs well in the older population. So this is sort of a matrix that you can apply. Actually, taking this one step further is to use machine learning to make those splits, to not sort of pre-specify what the matrix looks like. For example, you can use a random forest to actually do that. And that's something that we've also done. We've done that in the collaboration with a great batch of students from Europe, from TPAT, and that will be presented in a few months at Page. Then going to the right side, model retraining is also something we are highly interested in, because then you can actually go beyond the existing models. So what you can do is just a simple refit of all the data that you have. And again, you can split it up in sort of matrix if you want to make a specific fit for each group. But what you can also do is something called SIS, which I'll explain in the next few slides, where you can actually do that on the fly, or sort of with every the patient coming in, you can actually improve your models. So what is SIS? This is something that we developed in-house. It's called a composite importance sampling. And it's basically, you can see it sort of here in the cartoon as well. You basically build up the likelihood of the parameters. And it works more or less like this, sort of highly abstracted. If you would like more details, there's another presentation that I gave a year and a half ago at Page. But the basics work like this, where you start with a prior for a specific model. So you have clearance, volume, etc., and also the uncertainty around those estimates. And then with every patient that comes in, so this is one data set or data point for a patient, you are actually able to recompute the parameter estimates for that model. And you can do that with every new data set for every patient that's coming in. So after 10 patients, you basically base the new parameter estimates on all those 10 patients. So we try this approach in practice, not prospectively, but retrospectively. We knew that for one of the models in our platform, we were not so happy with its performance. This was a model for gentamicin in neonates, and we thought that the model was pretty biased. And we could easily see that, and of course this was retrospective. So this is the starting value for clearance, a little higher than 0.2. And this is the starting value for volume, a little lower than 1.5. You could easily see that even after throwing maybe 10 or 20 patients at this algorithm, it immediately signaled that the best value for clearance was most likely much higher than sort of the prior value. And over time, it really stabilized to a value of almost twice as high. So there was a huge bias in this model, at least for the patient population that we used it in. So this is just looking at the parameter estimates. We also computed the improvement in predictive performance. And also there we can see that the bias, if we had used this approach at the point of care, the bias would have been reduced immediately from over 150% to less than 50%, and also the RMSE it basically dropped 25-30% over time. So this would have been a really good application there. But here we have to admit we already sort of knew that this model wasn't performing well. So it's perhaps not a completely fair example. Besides using all the data that you have, what I showed before, just increasing the amount of data that you use, what you can also do with this algorithm is making a conditional estimate. And there you, for example, say, okay, I'm now seeing a patient that has a BMI over 30. Why don't I only recompute the parameter estimates based on these patients that I saw before that were actually over 30? So this is something you can do on the fly. You don't have to retrain the model, and that is something that we're looking at now, how that also performs. You can do it for BMI, you can do it for renal function. You can also actually join those two together. That's very easy to do. So in summary, for CIS, this is something that we're really actively working on. You're able to learn parameters from data streams versus doing everything in a batch. It's efficient and scalable and federated, meaning that if you had to, you can also run the algorithm at a specific hospital if they actually don't want to share their data, because you don't actually need the data. You only need to share back the likelihoods that you compute. There is a caveat that we think this approach is mostly limited to fairly simple models, and you can only re-estimate maybe 3 or 4 parameters, but that might be okay for what we want to do with this. So next, what I showed before is data cleaning here, and this step is very important, but it's not the whole story, because we do data cleaning here too, but we also do it before. And that's not something that we do. That's actually something that the user does, because our user is usually a clinical pharmacist or maybe a doctor. And when they receive the data from the hospital system, or they manually enter the data, what they do is they actually check if it looks okay. And if it's not okay, they may actually get a PK curve that's not doing well. They get warnings that the fit is wrong. So and in response to that, they can then remove a dose that was not actually given, or they can add a dose that was missing from the data. So the user is actually doing a lot of curation of the clinical data, and that's very important. But obviously, that's not enough. We still need to do some more cleaning if you want to use this for improving the model. And what you normally do, and I'm happy to hear other opinions on this. What I do, and what I've heard from a lot of people, what they do when you do a modeling analysis by hand, you basically fit an existing model, then you calculate the weighted residuals, and then you basically look at, okay, which are the worst points, the worst observations, and the worst the predictions, and you look specifically at these points. And maybe there was a unit mistake, or maybe the sampling time was wrong. It could be that the timing of the dose was wrong, something with the covariates. So there's all kinds of mistakes that happen. And yeah, if you can find those, you can obviously make a fix, or if you're not sure, you can remove the patient from the data set. But that's a manual process, and we were thinking, okay, how can we make this more automated? So together with my colleague Jordan Brooks, we started thinking about how to make this in a more automated process. And it is good to keep in mind that of course it's easy to just throw out the points that are not doing well. So if you have a cutoff of the weighted residuals being higher than 3, just remove those. That's of course easy to do, but you have to be careful because this is clinical data, real world clinical data, and there are just very extreme patients sometimes that actually change a lot over time, that are just that there's a lot of weird stuff sometimes going on in hospitals, that just that you actually see that in the data, in even when the data is actually true. So you don't want to remove those in principle. You only want to remove actual data entry errors, and that's what we're sort of trying to do here. And one approach that we came up with was where you start more or less the same as you would do manually. You fit an existing model, then you rank the observations by the residuals, and you throw out just one row, the worst predicted observation. You only remove that from the data set. So with that new data set, you then refit the model. And again, you throw out the worst performing row. And you repeat that process maybe 20 times, 30 times. Yeah, that depends a bit on how clean your data is and how large your data set. But you let's say you do this 20 times. During those 20 times, you also collect the individual objective function value. And that's important because at the end of this process, you then actually reconstruct the objective function value for the whole data set, but only looking at the patients that are sort of in the final data set. Because when you want to compare objective function, it has to be on the same data. So that's important. And if you then make a plot of the objective function over those iterations, you can see that going from the original data set, we set that to 0. Of course, if you remove the worst observation in that whole data set, you will see usually a pretty large drop in objective function value. But this means that removing only a single point improves the performance in the remaining data set by more than 30 points. So that's really a really bad point, most likely. Then over the iterations, you can see that, of course, the objective function value is increasing because the fit will get better because you're removing the worst points. And at the end here, between 20 and 30, it's really hard to say if those were actually errors or maybe just a bit of a weird PK point. But between, let's say, iteration 1 and 10, those are most likely wrong data points. Perhaps you can even remove them without looking at them. But especially in the middle area, this is where you probably still want to have a look at these points. And this plot can sort of help, to have a feel for how clean is my data, where should I look, how many observations should I remove from the data. And another plot that can also help with this is the parameter estimates over these iterations. And in this case, you can see that actually the PK parameters themselves, clearance, volume, etc., they did not move so much. it was only the residual error that was reduced from 2.5 to 1, which is a massive reduction. And of course, when you remove if you remove the worst data points from a data set, even if it's sort of a clean data, that will go down. But this is pretty dramatic. You wouldn't expect this large of a drop. So at least I would say the first 10 points that you should most likely remove those. And you could also see you can also say, like, why do we care actually about the residual error? Because if the estimates, the PK estimates don't actually change, is it really important? And I think it depends a bit on the application, but for our application, this is really crucial, because when you do a Bayesian update, that update is basically scaled by the residual error. The higher the residual error, the less it will actually listen to your data. So for us, that's a very important factor. So we really need to work off of clean data, and this is one approach that we're working on to make this more efficient. So in summary, I showed you.
Please wrap up.
Yeah, thank you. In summary, I showed you that there's many approaches to learning from your data. I just showed you a few that we are working on, but there are more. Data cleaning, I showed you, is important but challenging. There's no easy approach to this, I think. The users of these platforms already curate their data to a certain extent, but we need to do more as well on the back end. So with that, I thank my colleagues, Simulation Plus, for hosting this webinar, and you for listening. And I'm happy to answer questions at the end.
Thank you, Ron. Very nice presentation. Our next speaker is going to be Dr. Sophie Stocker. She is a senior lecturer at the School of Pharmacy in Sydney University, and also she holds an honorary senior post-hospital scientist appointment in the Department of Clinical Pharmacology and Toxicology at the St. Vincent Hospital in Sydney, Australia. Dr. Stocker has experience in industry, healthcare, and academia, and her research focuses on understanding variability in response to medicines and how these can be managed to optimize patient care. She is internationally recognized for implementation of precision dosing software and other precision medicine approaches. Dr. Stocker has co-authored more than 120 papers and holds several committee positions in national and international societies. She has gained recognition for her leadership internationally and nationally, and received several awards. Dr. Stocker, thank you for being with us today.
Thanks, Anna, for that kind introduction. Let's share my slide. So I'm going to talk to you about the implementation of model-informed precision dosing in routine clinical practice. And this talk is really designed to stimulate thought and discussion around the factors to consider when implementing and improving particularly a therapeutic drug monitoring service that utilises dose prediction software. And I'll do my best to cover most of the institutional and healthcare professional related factors that are listed on this slide. And my journey really is with the antimicrobial vancomycin, for which therapeutic drug monitoring has been recommended by clinical guidelines. And I've principally involved in the implementation of model-informed precision dosing service at St. Vincent's Hospital, which is in Sydney, Australia, and now also implementing this in hospitals elsewhere in Sydney. But just to orientate you a little bit, St. Vincent's is a relatively small hospital. It is a transplant, mainly heart and lung transplant referral centre, and also an ECMO referral centre for the region. It has an antimicrobial stewardship team that consists of pharmacists as well as clinicians. It's got a pathology service that's on site. It's on here on level 6 of the hospital, and they provide vancomycin concentrations 24 hours a day, 7 days a week, with a maximum turnaround time of around about 90 minutes. The hospital has an electronic medication management system, although ICU, their system is slightly separate from the rest of the hospital. As many of you will be aware, therapeutic drug monitoring of vancomycin is recommended, and that's because vancomycin drug exposure has been associated with not only the efficacy of vancomycin, but also the risk of toxicity. And here we're mainly worried about the incidence of acute kidney injury. So international guidelines have recommended that we transition from what we were doing originally, which is trough-guided dosing of vancomycin, to AUC-guided dosing of vancomycin, because AUC is a better predictor of both efficacy and toxicity. And the simplest way to do this is through the use of Bayesian forecasting software or dose prediction software. So it's probably not a surprise to this particular audience, but what we often use is this Bayesian approach where we can use patient characteristics. If we've got some vancomycin concentrations, then we'll use those to predict drug exposure, so an AUC in an individual using a population pharmacokinetic model. And then from that, we can predict what would be the most optimal dosing regimen in order to achieve the target drug exposure. And we can also play in silico with changing different dosing regimens or dose routes of drug administration as well. But before you start using a dose prediction software, you of course need to understand what's the most appropriate model, and you also need to understand the impact of factors on the accuracy of these predictions. So the precision of model-informed precision dosing really requires accurate information. So particularly the time of the vancomycin concentration, the time the vancomycin was administered, and as Ron has indicated, the appropriateness of the population pharmacokinetic model that you're using to predict drug exposure. And as you can imagine, in a busy clinical environment in a hospital, the quality or the accuracy of some of this information may impact the dosing decisions that you make. So we were very interested in this, and so we sought to better understand current clinical practice with respect to the collection initially of blood samples. So at St Vincent's Hospital, most of the therapeutic drug monitoring samples are collected by phlebotomists. So we followed these phlebotomists around using a time and motion study approach, and we also conducted focus groups with the phlebotomists. So what we found was the phlebotomists are very diligent and accurately record the time of sample collection. But due to a high workload, their inability to access the electronic medication management system, and a lack of knowledge regarding therapeutic drug monitoring, they actually aren't, it's not communicated to them that they need to collect a particular timed sample. So the sample collection from phlebotomists is essentially opportunistic and really relates to their workflow and work processes. So this was a really interesting finding, and particularly if you're restricted to collecting samples at a very specific time in the dosing interval, clearly using routine samples collected from phlebotomists may not be fit for purpose. We were also interested in understanding the accuracy of time of drug administration. So we reviewed the administration of more than 600 antimicrobial infusions. And essentially what we did is we have smart pumps at the hospital, and smart pumps have got a brain inside them. And we downloaded the time and event log from those smart pumps and then compared it to the time of drug administration documented in the electronic medical record. And we also interviewed nurses about their perspectives on local IV medication administration policies, and factors contributing to errors in the administration of IV antimicrobials. And what we found when we compared these two time and event pieces of data is that there was on average a discrepancy of 16 minutes between the time documented in the EMR of drug administration and the time the nurse pressed start on the pump. And some administrations had a discrepancy of greater than 60 minutes, where the nurses would indicate they'd administered the drugs, but when they went to the bed, the patient wasn't there. They were down getting an MRI, for example, and then there was a significant delay between them actually administering the drug. And when we looked specifically at vancomycin, we saw that this difference in the administration time actually had the potential to result in a different dose recommendation in a significant number of instances. So really highlighting the importance of accurately documenting the time of drug administration and communicating the importance of this to the relevant staff that are conducting these activities within a hospital. So as Ron has already touched on, selecting the most appropriate model is really also very important in order to ensure the accuracy of your prediction of drug exposure. And we've done less extensive work than Ron and his team, but we've done a little bit of work, at least with vancomycin, where we compared the performance of various models and also different dosing prediction software. And what you can see from here is when compared to area under the curve determined from the trapezoidal rule, there are definitely differences in the performance of models, but also in the performance of samples collected at a specific time. So there are areas within the dosing interval that are more informative than others based upon the description and the quality of the population pharmacokinetic model that's used. Interestingly, there are also differences in some of the weighting functions that are used in the various software that can also influence the accuracy of these predictions. And with colleagues such as Sebastian and Wicha, we have evaluated a range of the several vancomycin models that are available. We've used some of our routine data and then compared both the drug exposure estimated from these different population pharmacokinetic models. And then we could make some recommendations as to which of these models perform better in particular patient populations, including the critically ill, as well as a heterogeneous population. We've done this for vancomycin. We've also done it for tacrolimus, and we're continuing this work with other antimicrobials. So in addition to these institutional factors, there are also healthcare related factors that are important to consider when implementing MIPD in your institution. So of course, vancomycin dosing and therapeutic drug monitoring involves multiple healthcare professionals and essentially behaviour change. So moving from trough guided to AUC guided dosing decisions. And so to facilitate this behaviour change and acceptance of dose recommendations made by a TDM service, we conducted an education campaign. So the design of this education campaign was informed by interviews that were conducted with health education experts from around Australia. And essentially we ended up providing didactic seminars to the relevant stakeholders, case presentations, one-on-one academic detailing and lanyards, and the education was tailored to both junior doctors that did most of the vancomycin prescribing in our institution, as well as senior doctors, so getting them on board and creating champions within the institution. So of course, communication is also key. There's no point going to all the trouble of predicting the optimal dose and then no one accepting or understanding the information that you've provided. And so with an expert in design technology and human factors, we worked with designing the dose recommendation report. And design technology really highlights that there are key aspects that influence understanding and acceptance of information. And through interviews with prescribers, we could identify that they wanted information provided in a really simple way. And a drug exposure graph showing trends over time, as well as model fit, so a visual was seen as important, as well as the dose recommendation in a prominent space. And although we think it's very important to highlight the AUC, interestingly, the healthcare professionals we interviewed really reflected a lack of understanding of the term AUC or drug exposure. And so they actually viewed this information a little less important. We also spoke to our prescribers of where they wanted the dose recommendation, and they essentially said they would like it to coexist alongside the pathology records where they had the vancomycin concentrations. So you can see here that we've just uploaded the PDF of the report, and it sits right here next to the vancomycin concentration. And if a dose adjustment is recommended as part of the service, then we'll augment this with a phone call to the relevant treating team to enable further discussion and the opportunity to negotiate the recommended dose adjustment. So once we did all this preliminary work and implemented the service, of course, we were interested to see whether the vancomycin MIPD service made any difference. So we evaluated the AUC-based or guided vancomycin advisory service, and we used an interrupted time series approach to do so. And the primary outcome of this study was the proportion of treatment course that was spent within the therapeutic range. And we also looked at secondary outcomes such as the incidence of time to target attainment, and as well as the incidence of AKI pre and post service implementation. So based on literature, discussions which actually are ongoing with stakeholders and the existing hospital infrastructure, we designed the TDM advisory service. This is really a centralised consultative service, and it's highlighted here in the figure, and here are the various processes. But briefly, all patients prescribed vancomycin are automatically enrolled into the service. A dose recommendation is produced, and then it's reviewed by a clinical pharmacologist before being provided to the treating team. And as I mentioned, if a dose recommendation is, or dose adjustment is recommended, a phone call will be made to the treating clinical team. This service was provided to the bulk of the hospital, excluding ICU, and ICU was provided with the service on a referral basis. So the service ran Monday to Friday, excluding public holidays, and on the weekend, clinicians use the old previous existing model. So using the interrupted time series approach, we could see that after implementation of the service in July in 2018, there was a slight delay in improvement, but essentially the advisory service improved the proportion of time spent within the therapeutic range for a course of vancomycin, as well as it increased the time to target attainment. So patients achieved target vancomycin concentrations sooner, and they maintained it for the remaining of the course of therapy. And these effects were sustained for at least half a year to a year. The service was also associated with a reduction in the duration of vancomycin, the course of vancomycin. Interestingly, we saw actually quite a big shift from patients who would previously have received a continuous infusion and a significant reduction in the proportion of courses administered via continuous infusion. And when we spoke to prescribers about this, they essentially indicated that difficult patients, so patients who were difficult to get within the therapeutic range, would be switched from intermittent to continuous infusion so that they had a better control over adjusting and achieving optimal drug exposure. We also reduced the time to the collection of the first vancomycin concentrations, and the service, while it reduced overall drug exposure post the service, interestingly, we saw that there was a significant reduction in courses of therapy that had therapeutic exposure to vancomycin. And encouragingly, in line with this, there was a reduction in the incidence of acute kidney injury. After implementing the service for some time, we conducted some additional interviews with prescribers and pharmacists, both who had and hadn't interacted with the service. And essentially what we found was that individuals who were more likely to acknowledge and accept the recommendations from the service if they perceived that it would be associated with more beneficial outcomes, if the report or the advice was accessible. So if they were phoned directly, then it was very easy to accept the dose recommendation. And then over time, they developed trust within the service. So trusting that the people providing these dose recommendations, that they were accurate and also appropriate for their patient population. So really, we have a very high acceptance rate of the dose recommendations produced by the service. So it's more than 85% of the dose recommendations are accepted. And really, this advisory service is now ingrained within the institution.
Sophie, we have a couple of minutes.
Hopefully I've been able to show you that understanding clinical practices and workflows and really engaging with stakeholders in your institution should inform the design of your model-informed therapeutic drug monitoring service. It's important for you to consider what model you use to predict your drug exposure and have a bit of an understanding of the accuracy of not only the timing of the vancomycin concentrations, but also the administration of drug at your institution. This particular model-informed precision dosing service, I think, showed improvements not only in therapeutic exposure in individuals, but also reduction in adverse effects. And obviously, further optimization of the service could further improve on these findings. I think using an interrupted time series approach when using real world data should be advocated. And I see more and more researchers using this approach when evaluating model-informed precision dosing services. Finally, I'd like to acknowledge all of the individuals involved in these various studies, the funding agencies, and of course, the organisers of this webinar and IATD-MCT, the opportunity to speak today. Thank you.
Thank you, Sophie. And we will go to the introduction of our next speaker, who is Dr. Sebastian Wicha. And he is a professor of clinical pharmacy at the Institute of Pharmacy of the University of Hamburg in Germany. Sebastian's research group aims to improve and individualize drug therapy with a special focus on anti-infectives. His group has expertise in bioanalysis, pharmacometrics, and develops open access web-based software to foster model-informed precision dosing at tdmx.eu. He has published more than 100 peer-reviewed papers in the field. He served as president between 2020 and 2022 of the International Society of Anti-infective Pharmacology, or ISAP. He is the education officer of the PKPD study group and fellow of the European Society for Clinical Microbiology and Infectious Diseases, or ESCMID. He's also an external expert for pharmacometrics for the German Federal Institute for Drugs and Medical Devices and the European Medicines Agency, EMA. So thank you very much, and please go ahead.
Yes, thank you, Amparo, for the very, very kind introduction. I will share my screen, and I hope you can now see my slides.
Yep, we can see them.
Fantastic. Okay, so very nice presentations already from Ron and Sophie, and we will go a bit further into MIPD approaches with anti-infectives and looking at some challenges and potential solutions. I do have a few disclosures that I'm quickly projecting here, and then let's start. So just a quick recap. Maybe a few things here are already very well known to the audience still. Why do we want to do model-informed precision dosing? What are the advantages over the conventional TDM approach? Well, we can already actually calculate a likely effective first dose even before we dose the drug. And thereby we can include, well, all sorts of covariates, and that's much more inclusive than label or guideline dosing where maybe one covariate is considered. Secondly, we don't have to wait for a steady state to draw a sample and already inform a model and do a dosing decision. So that is particularly important for anti-infectives where speed is life in sepsis, where you want to have therapeutic concentrations as early as possible. And then also you're very flexible with the timing of the sample. Any timed sample can be used. You don't have to specifically time, for instance, for a trough sample. And then also from that single sample, one can obtain an estimate of a full PK curve. And that is also advantageous because once we have the individual PK parameters, we can target a specific exposure metrics, time above MIC, an AUC target, as we heard for vancomycin, and we can really try to find a dose that is in the middle of the exposure range, whereas the traditional approach where you just compare to a target, you probably do not reach the sweet spot to be at the middle of the target. So we can maximize the probability of target attainment on the patient level. And potentially we could include all sorts of biomarkers also. There are some models also available that, for instance, can link CRP time courses together with PK, although this has not reached the step to be implemented in practice yet. But I think maybe in the future we might see also models that include those biomarkers and can thereby also help in the decision-making of the therapeutic course by not only looking at PK, but also at PD. But there are also, of course, some challenges. We have heard that already a little bit. Which population model shall we use? Also, when to sample. In the field of anti-infectives, it's particularly problematic that you deal with unstable patients where the PK is, deteriorating over time. In this webinar, I will also go a bit into the direction of which population model to use and show some of the work we have done in our group and also some solutions we think we have also, or our students also have tried to contribute. So for anti-infectives, which model to choose? Maybe we start with a few pictures. So you can imagine you have really various patients in the hospital. You may have general ward patients that are, rather healthy but suffer from a specific infection, for example. But you may also have this situation on the intensive care unit where patients can be very sick, suffer from critical illness, which significantly can affect the volume of distribution. You may have organ failure or augmented renal clearance. You may have organ support. So really a number of factors that certainly have can have a drastic impact on the pharmacokinetics of anti-infectives. And then sometimes you have even a combination of problems here, for example, a critical ill patient that is also obese at the same time. So a very heterogeneous cohort overall. And also then the big question, which model shall I actually use to do TDM in such patients? And a few years ago, when we implemented vancomycin into TDMX, we did a literature search, and we really found a huge number of vancomycin models that were available. It was more than 40 at that time. And also when, just simulating with these models, we really realized that they displayed a very heterogeneous predictive performance. So we thought we really need to systematically evaluate the predictive performance of such models before we implement them. And here we looked at the general ward patients, ICU patients for the intermittent infusion setup, but also in the continuous infusion setup, which is also popular for vancomycin. And we did that together with Sophie and also with other colleagues in Europe here with Peter de Kock from Belgium, and also colleagues from the United States. So how did we evaluate the predictive performance in those data sets? We tried to mimic the TDM process retrospectively. So we used one so-called reference occasion, which we used to, evaluate the predictive performance. And we tried to predict these data points, but they were hidden from the models. And then we tried out different setups. So the a priori setup, where we would just have used the dosing record and the covariates, but no TDM measurements. Then, different variants of Bayesian forecasting. For instance, here where the first observed occasion was used to predict our reference occasion, or here the most recent one. So to also evaluate a little bit the impact of time and age of the sample, and also here by adding more and more data points to see whether adding more data can actually also improve the predictive performance. And then the a posteriori, the Bayesian estimation, where we included all data points that were available in that patient. And actually, before I show you the results, this is a very interesting paper that looked at the evaluation of model evaluation. So we were here, that's the paper from us. We were not the only ones that looked at external model evaluations, and they were actually evaluating what criteria were used to evaluate those models. And you see here all sorts of things, and the analysis I just explained to you is the Bayesian forecasting analysis, which we think is actually quite important, because this is the domain and the setting how these models are being used in the TDM setup. And you see a lot of red circles here. So many of these evaluations actually did not look at this Bayesian forecasting performance. So also maybe very important when you think about how those models are evaluated. Here, let's look at some results for vancomycin for the intermittent infusion general ward cohort. You see here the different scenarios that were evaluated. You have here the bias and the RMSE. And as you can see, most of the models are actually improving once TDM data becomes available. Also, the most recent dosing occasion is more informative than the first dosing occasion. And actually, adding more data does not necessarily always help. You can also see that the predictive performance of those models was very heterogeneous. Sometimes we could tell it was a very different population sometimes, but sometimes maybe also there was maybe a mistake in the reporting of the model, or also sometimes models were only developed on trough data, so they would probably not predict non-trough concentrations very well. Nonetheless, we found one model that performed best, the model from Goti et al. Here. Then we went to the next data set, and that's now the continuous infusion scenario. So it's the same general ward cohort, but just a different mode of administration. And then if we go again to the Goti model, which we which we can see here. Well, now in just by changing the mode of administration, you see that this model is actually not the best model anymore. It displays some bias. And two other models, the models from Okada and Collin here, did perform best. Well, and that was a bit disappointing for us to see, okay, if you just change one more small thing, you already end up with different models that work best. And we are already thinking about a decision tree that we could add to our software. But we found that this all was not very practical, and we stepped back and tried to come up with a more user-friendly solution. And therefore, we came across the work from Yasunori Aoki, who was a postdoc in Uppsala at the time, who looked at new approaches for phase 3 dose selection. And traditionally, when you come up with a dose selection exercise, you have your data set, you fit different exposure response models, and then you will find that one model may perform best here based on Ikaiki criterion, and then you use this very model to do a simulation. And Aoki et al., they then came up with a model averaging approach that you actually do not only use a single model for making these predictions, but you actually use all models to make these predictions and then weigh the predictions according to the model fit. So a model that has a very low model fit gets a low weight, and models with a good model fit, they get a very high weight in that prediction. And in their exercise, they could show that they could actually improve the predictive performance and the certainty in the dose selection. So we try to use this model averaging approach and for the TDM setting. So how would that work here? We would take one patient, and instead of fitting a single model, we would fit a number of candidate models, which is depicted here. And then we also would weigh those models after the estimation step. The better models would get a higher weight, and the models that do not fit on the individual level very well, they get a lower weight. In this example here, we use the objective function value to calculate these weights. And then in the next step, we can do two things. We can do a model averaging, which is that black line that is added here. So where we would calculate an averaged PK profile across those candidate models. Or we could also do something like winner takes it all, and the model with the highest weight is selected and the other models are discarded. So a more simple model selection approach on the individual level. And to set up this algorithm, we used a number of very different models. As you can see, these are really models stemming from different populations. And also, if we would simulate a standard patient with the same covariates and the same dose, you can see that these models also generate quite different PK profiles. Now, we used this approach first in a simulation study, and we wanted to see whether the algorithm can find the simulation model again. So we have here a number of models that were used for simulation, and then here we see the percentage, the distribution of the highest weight for each model. So we would so a priori, you see all the weights are the same, but once we add TDM data, we would want to see that the fraction that is found to describe the original simulation cohort best should become bigger, and that's what we see here. So here, for instance, this pink bar gets larger, and also here. And the more data you add, the more are the weights dominated by the simulation model. So that's nice. So the model can actually detect the true model in the simulation study, and also the forecasting performance could be substantially improved. So you have here with these different shapes, the simulation models, And then here on the x-axis, the estimation models. You have here the bias and the RMSE for accuracy and precision, and here for 1 and 2 occasions. And you can see here the model averaging algorithm really produces here the lowest overall imprecision, and also with regard to bias looks pretty good. But this is a simulation study, so we also wanted to see whether we could translate this finding in a real data set here. 180 patients here in this data set with a mix of general ward and ICU, so a very heterogeneous cohort. And we have here again bias and RMSE here adding more data, and we have here the performances of the individual models, which can be quite heterogeneous with positive and negative bias, and then the averaging algorithm, which was unbiased, and also the imprecision was usually in the range of the best performing model. And also we found here that the performance was better than the one from the GOTI model, which we previously identified to work actually pretty well. So we also looked at the robustness of such an algorithm. Can we actually, how many models do we need for model averaging? And here we are, from here to there, we always remove the best performing model from the set of models here. And actually, well, it still works pretty well, even with only 3 models. Then if we went down to 2 models, you see that the predictive performance got significantly worse. And the model selection algorithm, well, overall performed worse than the averaging algorithm. And what I can mention is what is really important for the model averaging is that you have candidate models that display a positive but also a negative bias, which was actually the case here in this vancomycin exercise. So the models can approach the data points kind of from both sides. Whereas if you have only positively biased models, well, then the averaging could get as good as the least biased model, but you will obviously never reach an unbiased situation. Yes, and that not only works with vancomycin, also with other drugs. That's a paper we did together with Erwin's group. I'm not going into the details because I'm sure we will hear more about infliximab, or here a coagulation factor, where the model averaging also performed pretty well. We also looked at different weighting schemes. So you have now seen the one with the objective function value. We also looked at Ikaiki as the one that was originally proposed for model averaging in the Aoki paper, or also the SSE, the sum of squared errors. So really the prediction errors and the extended least squares, where we also considered the residual error. We have to look here in this left column, the one for Bayesian forecasting. And we can see here the performance of the single models for model averaging. The differences are actually very small between those different weighting criterions, but overall objective function value and SSE tended to perform best, and Ikaiki performed not so well. Can imagine why. If you penalize models that have more etas to estimate, that's probably unfair, because I think Ikaiki is probably not the most accurate criterion here to evaluate the model fit on the individual level using a Bayesian estimation approach. If you want to try our algorithms out, you can do that on the TDMX website, which is available open access. If you want to use a single model, you are in the single model tab here. If you want to try model averaging, you can tap on the model averaging, and then you can see here the color coding gives you the different weights of the models, and then also here the averaged prediction, which can be seen here. So I want to wrap up. As you know, available pharmacometric models are very often built actually not with the use case MIPD in mind, which also means model predictive performance can be very heterogeneous, and that's not only true for vancomycin, but also for other external evaluations we have performed so far. Novel algorithms can improve the predictive performance, such as using some automated model selection or model averaging, and even combinations of models that individually perform bad can actually then result in a good predictive performance in the model averaging setting. But we also need more implementation research. That's my experience, that training of healthcare professionals is really important, and that has to start already in the undergraduate level, that these approaches are taught. And then also we need more comparative studies, implementation studies, such as those nice examples shown by Sophie, the previous presentation, where you compare MIPD and show also the advantages of MIPD approaches in the real world. Yes, and here I just want to acknowledge again Sophie, Peter, and Ken Klinker, who donated the data for the validation studies I have shown today, and also Astrid Bröker and David Uster, who were PhD students who were conducting some of the studies that I've shown today, and of course our funders for making that possible, and also the organizers of the webinar today, Simulations Plus and IITD-MCT, for endorsing this activity. And I'm happy to answer questions at the end.
Thank you so much, Sebastian, for such a nice presentation. Our next speaker is Dr. Erwin Dreesen. Dr. Dreesen is a full-time assistant professor and head of the pharmacometrics research group in the Department of Pharmaceutical and Pharmacological Science of KU Leuven in Belgium. His research group has a specific interest in model-informed precision dosing of monoclonal antibodies, anti-infectives, and immunosuppressives. At KU Leuven, Erwin teaches courses on applied pharmacometrics, clinical drug development, and model-informed drug development. He is a member of the Pharmacometrics Scientific Committee of the International Association of Therapeutic Drug Monitoring and Clinical Toxicology, the Ethics Committee Research at the University, and an expert for the Committee of Medicinal Products for Human Use of the European Medicine Agency. Without further ado, Dr. Erwin, please, you can proceed.
Thank you, Anna, for the very kind introduction. Let me try and share my screen. I think that worked.
Yep, we can see it.
Perfect. So hello, everyone. After these very nice talks about using real-world data, implementation strategies, MIPD of anti-infectives, this is what I will bring to the table. I will share some insights into MIPD of monoclonal antibodies, and this in patients with immune-mediated inflammatory diseases. So these are some disclosures that I have. Now, let's start with a more existential question. Why are we in fact talking about MIPD of monoclonal antibodies? It is a very easy to answer question, simply because there is a huge problem in the clinic. Although these monoclonal antibodies are very effective drugs, they are only effective in a minority of patients who receive them. And that is what we can see in this figure. So it's just an example. Here we're looking at the remission rates in the pivotal ulcerative colitis trials, showing remission rates of less than 50%, and often less than 20%, in fact. So this is what we call the therapeutic ceiling. Question is now how to break it. And this is something clinicians are looking into. They try different things, combination therapies, predictors of response, switching between drugs in the expanding armamentarium, and of course also TDM, therapeutic drug monitoring. And several years ago, we did a survey and a good comparison of all the available MIPD software tools available at the time. And it was already very clear that after anti-infective drugs, monoclonal antibodies are an important class of drugs covered in many of the existing MIPD tools. So let's dive a little bit further into this. Here we have the options that we have today. So if a clinician or a clinical pharmacologist would like to implement MIPD of a monoclonal antibody into their clinical practice today, 6 software tools out of 14 available allow us to do so. For which drugs? That is something I will show here. So you can see that some softwares have a lot of drugs, but there's also a clear top 3. Infliximab is available in all tools. Then we have adalimumab available in 4 out of 6 tools, and vedolizumab in 3 out of 6. So there's a very simple reason for that. These are really the most widely used and also most studied monoclonal antibodies in these diseases. So infliximab and adalimumab, just as a side note, are anti-TNF drugs. Vedolizumab is an anti-integrin drug, specifically targeting the bowel. That's why it's used in gastroenterology and inflammatory bowel disease, Crohn's disease, and ulcerative colitis. And the anti-TNFs are used also in rheumatological and dermatological inflammatory diseases. So great, we could actually just start doing MIPD with these monoclonal antibodies, but we need to take a step back. We need to think, what is the actual evidence? It is not because there is a software tool that there is also evidence for its benefits. So looking into the evidence of MIPD, I'd like to generalize it a bit further and also look into the evidence for model-based TDM or MIPD or TDM in general. And then it's pretty simple. We have most evidence for infliximab, less for adalimumab, and even less, or not to say almost nothing, for vedolizumab. Most evidence for infliximab. What does that mean? It's easy to summarize. It's really only 5 randomized controlled trials performed over roughly a decade. And on the left, you see the 2 clinical trials that are actually able to show benefit of TDM. So there's, of course, a lot to say about these trials. I'll keep it short here. I think the main takeaways are that both trials showing benefits of TDM focus on TDM in the maintenance phase, which means when patients are already on infliximab for several months. The difference is that the oldest study, the Stenold study, 2014, that it focuses on reactive TDM, which means performing TDM in patients who lost response, trying to restore the response. While the Nordstrom study, the most recent study, focuses on proactive TDM, which means doing TDM as part of routine care, trying to avoid a loss of response. Of course, we have to keep in mind that between these 2 studies, 3 other studies were published that were not able to show benefit of TDM in the primary endpoint. There's many reasons for that. I will highlight one a bit further in this presentation, but I think it is clear that we started with high hopes about 10 years ago, and now we are very slowly regaining hope for TDM of infliximab thanks to the Nordstrom trial. This trial really showed that performing TDM was able to help clinicians to better control the disease of their patients. And of course, clinicians, they need to make sense of all of this, of the positive trials and negative trials. And it was a pretty bumpy road. And over the years, they tried to derive some evidence-based actions, which were published in guidelines. And this was mainly a national affair, as you can see here. Many guidelines, not always drawing the same conclusions. But if you would look into the more recent guidelines published after the Nordstrom trial, there's a consensus that there is a clear benefit of TDM and that there is better hope. Looking into the Nordstrom trial, the trial that actually showed a benefit for TDM, as you can see here, in this trial, they were not able to bring all patients within the therapeutic window. Therapeutic window from 3 to 8, which is, by the way, a very wide therapeutic window. And after 2 months of TDM, there were still a lot of patients above and below the therapeutic window. And even after 1 year of TDM, not all patients were within the therapeutic window. So this is obviously an opportunity to do better. And maybe this is also a reason why some of the previous studies that I showed, the Taxid and the Tailorix trial, were not able to show benefits of TDM. So we need to look into the algorithm that was actually providing the dosing recommendations, and that is what we see here. I simplified it a little bit for you, and it is actually very simple. If you're below the therapeutic window, you increase the dose or you shorten the interval. If you are above the therapeutic window, you lower the dose or you extend the interval. Now, this is of course not precision dosing. This is trial and error, and over the weeks to months, step by step, you bring your patient within the therapeutic window. In the Keizer trial, a trial that was not able to attain a primary endpoint, a very similar algorithm was used. It was visualized as this flow chart, and clinicians, of course, they like these algorithms. They are simple, easy to implement, and even though the Keizer trial did not attain the primary endpoint, every clinician knows the Keizer algorithm and basically applies it in clinical practice if the regulations allow to. Problem is, it all works very slow. So room for improvement, and this is of course where MIPD comes in. MIPD at least promises to do TDM faster and more precise. So let's look into this. And in fact, several years ago, this study was published showing the benefits of model-informed precision dosing of infliximab in IBD patients. So that's great news. In this study from Amsterdam, they showed that patients on infliximab maintenance therapy dosed following MIPD targeted at a trough level of 3 milligram per liter had a longer remission rate. Well, so to say, after 1 year, they had a higher remission rate as compared to patients who just followed standard dosing. This is something they saw in the per protocol analysis, but not in the intention to treat analysis. How come? Well, because of a number of patients in the intervention arm, in the MIPD arm, that had a dose de-escalation, trying to bring them to a level of 3 milligram per liter. And by doing so, they had a reopening of their old fistulas. This is a very interesting observation, in fact, because this made us wonder, are we in fact targeting the right trough level? 3, is this the right target? Or maybe, probably, some patients, especially these patients with fistulizing Crohn's disease, may need more infliximab, may need higher trough levels. So what's left in literature? Not much. This is one other interesting paper, I believe, group in Cincinnati, describing the development of a software tool for MIPD of infliximab. And what is specific here is that they also describe the integration into the electronic health records in the hospital. And I think, of course, that's important. That was already highlighted before by Sophie, because TDM, of course, is a burden in healthcare, and we should try to make it as user-friendly as possible. This is really it, but of course there's more to come. And for that, I would like to just highlight the titrate study, a study that was performed in Amsterdam. And there's 2 reasons why I really like this study. First reason is because it focuses on very vulnerable patients, patients with acute severe colitis, patients that actually might benefit from TDM. There's a lot at stake, there's a lot to gain. Another reason is that in this trial, they used point-of-care assays. So this means they used assays to measure infliximab concentrations with a very short turnaround time. Results are available in 30 minutes, and dosing can be optimized immediately. And I believe that besides MIPD, of course, it is all these things together that will be able to make model-based TDM or TDM in general more successful. I think it should be clear now that we are still in the research and development phase, and it is not yet time to just implement it in routine care. So we need to work on improving TDM, we need to work on improving model-informed precision dosing, and in fact, we should wonder how precise precision dosing is. Because if you look into the algorithm that was used, the model-based algorithm that was used in the precision trial, you can see in this small plot that after 6 months of MIPD, not all patients were exactly at the target. There is variability, and in fact, the mean was just above the target. So also here, at least it requires investigation and maybe an effort to improve things, trying to bring all patients to the target. And for this, we were much inspired by the work of Sebastian in Hamburg on vancomycin. So what we did is we developed a multi-model averaging and selection algorithm together with Sebastian for infliximab. This was work of my former PhD student, Wani, who coded all the 18 infliximab PK models, built this algorithm, and validated it retrospectively. So what happens is that we feed 1, 2, or 3 concentrations into the algorithm, predicting a future concentration, and then comparing this predicted concentration with the observed concentration, calculating bias and precision, and doing this for a data set of over 60 patients and summarizing it for all of the models. So here you can see that most of the models, of the 18 models, were biased, usually a negative bias, and the precision was also not very good, roughly around 30%. The multi-model algorithms, at least the model selection algorithm, was unbiased, but the precision did not really improve that much with the 18 models. So we worked on the algorithm, trying to make it as unbiased and precise as possible for these Leuven data. And that brought us to this algorithm with 4 models. 4 models, that also means the algorithm is not too time consuming and actually workable. And this algorithm has been implemented into TD metrics. We developed an algorithm for adult patients, but also in a pediatric context. And now, of course, the next step is to validate the algorithm prospectively in a clinical trial, which is exactly what we're doing at the moment in the modified trial. So to end my presentation, I would just like to present how all of this works in this clinical trial. So here we have patients who are on infliximab, and at some point they lost response, which is why the clinicians increased the dose or shortened the interval to have them give them higher exposure. And this was successful for many patients. They all brought back into remission. But now, after months and years on escalated regimens, the question is, can we again de-escalate, bring these patients back to standard dosing, of course, in a safe way without risking relapse? Now we are using MIPD to do this. So this was the first patient, the female patient, 63 years old, with ulcerative colitis, doing perfectly fine, completely in remission, based on clinical scores, also based on lab parameters. And this patient had an infliximab trough level of 7.2. The dosing interval was every 6 weeks instead of every 8 weeks. So now let's bring this patient back to every 8 weeks. We used the TD metrics module from infliximab. We provide all the patient characteristics, the necessary lab values for the models, the historic dosing information, and then we click the auto select button, and what happens is that the best model is selected and visualized. This happens to be the model by the colleagues in France, in Tours, the model of David Ternon, and this was the best fitted model for these data, which you can see here. There's also the model averaging algorithm, which you can see here. And the funny thing is here that this one model by Ternon actually fitted the model so well, much better than the 3 other models, including my own model, which is a pity, of course. But this one model was so good that it took the entire weight. The other models were not contributing any whatsoever. So actually here, there is no difference between model selection and model averaging. And then this is why we continued with the model selection. We asked the algorithm to calculate a new infliximab dose that we should give to the patient, bringing the patient at a trough level of 5 milligrams per liter. That was our target, but this time 8 weeks later. The calculated dose was 510 milligrams. We accept the dose. And here you can see the predictions, which is a trough level of 5.4 milligram per liter 8 weeks later. 5.4, not 5. This is because we are also aiming for at least an 80% probability of target attainment. Now, I wish I could show you how well we did, but for this first patient in the first sample, the sample was lost. So this is very, very unfortunate, but this is real life. We learned from it. We continued with the trial, so please hold on. We did another dose calculation, this time 200 milligram. Software predicted trough level of 5.5, this time with a probability of target attainment of 88.8. We accept the dose, the clinicians administer the dose, and a trough level was observed this time of 4.8 milligram per liter. Now, 4.8 is below 5, so you might say this is not good, but in fact, 4.8 is actually 5. It's within a 95% prediction interval. It is also within an acceptable range of lack of precision, considering the assay variability. So in fact, we are very happy with this concentration. And today the patient completed the trial. The patient is in remission at dosing intervals of every 8 weeks, and in fact today longer dosing intervals, so less visits, better for the quality of life. And the patient is always at a trough level of 5 milligram per liter. So now recruitment is over. All patients are in the trial. We're at full speed, and hopefully later this year I can show you the results of this clinical trial. So my take-home messages. I think the most important thing is that research comes before implementation, and we are still in the research phase. Clinical trials are ongoing, some of which I did not discuss in detail, but please believe me, in the next few years, we will have more clinical trials, hopefully showing the benefits of MIPD. It doesn't mean that the availability of a software tool means that there is clinical evidence. And of course, as was highlighted in the previous presentations, MIPD is only one aspect of dose optimization. We need to do this in the right patients, the most vulnerable patients, where there's a lot at stake, but where, of course, there's also a lot to gain. We should also consider patient preferences, quality of life, especially for these drugs. The target, I've only been talking about trough levels, but recently a discussion has started about AUC targets. So maybe for infliximab, we are going towards a scenario like vancomycin. And other opportunities have been taken, for example, also to look into treat to target the clinical outcomes. Maybe in the future, we are using PK/PD models to do MIPD of infliximab. The data, I briefly highlighted it. We have better assays with a shorter analysis time compared to the ELISAs, which will allow point of care TDM. Also, biomarkers will be very useful in the future for PK/PD model-based TDM. And in the end, all of this, of course, needs to be implemented in clinical practice in a user-friendly way, in a cost-effective way, and of course, considering all the regulatory concerns that we have today, which is the challenge these days, also if you want to do research. So with this, I would like to end. I hope I give you a quick overview into what's going on in the field of TDM and MIPD of infliximab and monoclonal antibodies in general. Thank you to the organizers for Simulations Plus, and thank you for your attention.
Thank you so much, Erwin. And without further ado, I'll introduce our last speaker of the day. His name is Dr. Manuel Ibarra, and he is a professor at the Pharmaceutical Sciences Department at the School of Chemistry in the Universidad de la República of Uruguay, and the director of the university's Bioavailability and Bioequivalence Center for Medicine Evaluation, or CEBIOBE. His research is focused on exploring and developing pharmacometric models in basic and applied pharmacological research, aiming to understand specific pharmacokinetic processes and translational applications, such as model-informed precision dosing, virtual and model-based bioequivalence, and drug product development, among other applications. Some of his work includes the local and regional active promotion and dissemination of pharmacometrics. Part of that is that he is a co-founder and coordinator of the Ibero-American Pharmacometrics Network, or REBIF. In addition, he is a member of the World Conference on Pharmacometrics, WCOP, Executive Committee in representation of Latin America. So, Manuel, please go ahead.
Thank you very much, Amparo, for a nice presentation. Hello, everyone. You can see my screen now.
Yep.
Thank you. I'll start. Okay, great. So very happy to be here, sharing a bit on what we have done or what we are doing here in Uruguay, implementing model-informed precision dosing in the clinical practice. And yeah, definitely great to be here sharing this great panel and Very happy to be here with you. Okay, I'll start. For those who don't have to know this. Uruguay is a small country in South America. 3.5 million people. We are located in Montevideo, which is the capital, and we're based at our group is based at the University Hospital, which is that huge building in that picture, right beside the Centenarian Stadium, which is the place where the first World Cup was held in 1930. So yeah, our group. So the University Hospital is the largest medical education institution in the country. A lot of specialized services and definitely a large institution attending a big population of patients. It has 350 beds, mainly attending adult patients, and we have a therapeutic drug monitoring unit, which was created in 1991. It was created by professors from our group, Professor Marta Vázquez and Professor Pietro Fasciolino. And it is, I'll say it is the only TDM service, like full service, providing TDM in the country. So in this hospital, our group that belongs to the university, and which I have the pleasure to lead together with Professor Cecilia Maldonado, we are a hospital-based pharmacometrics group. We have been working with integrating pharmacometrics since some years now into the clinical practice, and we're in charge of the TDM unit. So we are slowly, because we think it's important to do it right, integrating more informed precision dosing in specific populations and specific to optimize specific treatments, critically ill patients, neonates, and renal transplant patients. And we are also working on more informed drug development, mostly in bioequivalence and control release formulations. But what I will share with you today is what our efforts are. It's our efforts on enhancing TDM with what it can be called as real-time model-informed precision dosing. Sebastian, which I already touched on the limitations and comparison between the classical approach to provide or to perform TDM versus the implementation of population PK and PD models to, I will say, enhance TDM. I think it's important to keep this say that we can improve or be more precise in what we are doing, but TDM is definitely the application on which we want to be of the application that we are part of, definitely. So very briefly, the real-time model-informed precision dosing will be the use of modeling simulation approach in combination with individual measured patient and disease characteristics to inform clinical decisions in the context of precision dosing. So mainly what we are doing is using population PK, PD models as to have a priority distributions that it's a way of integrating information, integrating knowledge, and then to combine that knowledge with what we can observe patient by patient, and using Bayesian forecasting, integrate observations with knowledge in order to have more precise predictions and projections on the individual pharmacokinetic outcome. And what I would like to touch on a couple, so 4 key aspects to be able of implementing model-informed precision dosing in an optimal way, in a successful way. And the first one, I think, is people. You have to think about the multidisciplinary framework that is needed to fully bring model-informed precision into the clinical practice. We have to bring in and discuss and interchange with very different healthcare professionals, and thinking with this in mind, we have created a group, an interdisciplinary group 3 years ago, with it integrates pharmacists, computer engineers, physicians from different specialties, veterinary doctors as well, and statisticians to discuss. And we have gone through a very very nice process of internal discussions discussing applications, discussing how we can implement this. I think we have, and it's important to be cautious about expectations that we can create in the clinical in the healthcare providers when we talk about bringing such tools into the clinical practice. So, and I think what Sophie just showed in her presentation has very nicely shown how we can study the implementation in a quantitative way, and really to really have measurements about the how this really improves the practice with different markers and so on. So definitely thinking in people and thinking how this is more a process, and we cannot perform more informed precision dosing from one day to the other. The other aspect, important aspect, is data, definitely, and previous speakers have touched on this before. So I think the some strengths that we have in our group, as a group that is in charge of the TDM unit, we have the opportunity to collect the data in real time and to assess the quality of the data in real time as well. And this implies analyzing the data, having the history of the patient, that if it's not a new patient, and compare and to see if we need to go to the bedside and have some discussions, engage in discussions, or with the nurse or the physician, see if we can go into the clinical history to complete the information, and so on and so forth. And we have also the ability to provide analytical solutions for accurate measurements of drug concentrations. These discussions with physicians have given us the awareness of important issues that needs of medical staff, and this definitely increase the impact that we can have on the hospital, bringing this tool into the practice. And we also are working on give timely response to physicians. That is definitely an important thing. The other important aspect is definitely models, and I will give an example on tacrolimus. So when we think about implementing MIPD into the clinical practice, there are mainly two options. One is implement your own model, your in-house model, built using data from the target population. If there is the opportunity to collect data or to have retrospective data and to develop your own model in the target population, but then there's also the opportunity to bring in published models as shown before, and also with different techniques, use those models with available platforms to start performing MIPD. What I think is very important and also have been highlighted by previous speakers is that we need to evaluate. We need a fit for purpose evaluation we need to evaluate the model before implementing it. So we need to validate the model in a target population. That is very important, and with some caveats that I will discuss later on. And the example I will bring in, it's a study that we are doing on tacrolimus. Tacrolimus is a immunosuppressant drug, and we are studying its application in renal transplant patients. Tacrolimus is a drug with a narrow therapeutic index, very high between subject variability and between occasion variability. And that it is used as an immunosuppressant. It's a first-line drug in the case of renal transplant patients and other types of transplant patients as well. here we show how so we first when we thought about bringing, so we are working together with the nephrology center here at the University Hospital, and when we thought about starting to implement IMIPD to optimize the treatments, the tacrolimus treatments, we start by reviewing the different models that were published. There are more than 30 models for tacrolimus in renal transplant patients, and here you can see the variability of those models for a typical subject and with the same covariates. How the different models predict the tacrolimus exposure throughout time for a typical subject, and as you can see, the variability is huge, and I think this is very natural and very common if we think about a drug with very high inter-individual variability, and that these different models actually represent different populations and different settings, different, of course, different modelers, but also different quality of data and so on. So we did a study in which we first studied in a retrospective manner. We studied 39 data from 39 adult patients. Almost 400 observations, and we did an external evaluation of 26 published POP-PK models. After reviewing all models and qualifying the models in different aspects, we defined that we wanted to evaluate those 26. And then we did a prospective prospective study in which 49 adult patients were enrolled, and then we developed our own model using this data set. And this data set was collected in a more, I'll say, a more precise way. So we put more emphasis on the time of data collection, the quality of the data. This is the work of PhD student Martino Pierres that was overviewing this. It was by the bedside discussing with the physicians and seeing the data in real time and enrolling patients. So the first thing I will show you is the standard evaluation. As Sebastian already discussed, it's important to evaluate the model in the way that it will be used in the clinical practice. So we first we split the data in different occasions. And we define that one occasion will be the curve. So it's a normal practice for tacrolimus to get a curve at some point after on the treatment, and the other sample points are mainly troughs. That is the most common type of measurements that we have. So we split this data, and we are evaluating the different models and in different occasions. And we first perform an a priori evaluation, and then we're integrating data and see to study how precisely are the data of the next occasion predicted by each model. And then the data from the second occasion is integrated and to predict the third occasion and so on. So here, and you can come up with different metrics to evaluate that. This is a relative bias, which is a metric for accuracy. Of course, it's very important to have precision also in your evaluation. So these are the 5 best models, and yeah, as you can see, there is these are the 5 best models, and we are here plotting the relative bias throughout the occasion, so occasion 1 to 5, and we saw that there is an improvement on the accuracy after integrating the curve into the data set, into the models. So and definitely one thing that you will expect. And this other metric that is very interesting is a metric that combines is that it assesses accuracy and precision. It is the proportion of patients for which the prediction error is below 30%, And here you can see more clearly the increment, so the improvement when more data is integrated into the model. Okay, but the most interesting thing it was when we developed our model, the model for tacrolimus, and the project was led by Professor Cecilia Maldonado and Oscar Novoa. We developed a model that integrated a free fat mass and hematocrit into the, and also the polymorphism of CYP3A5 as important covariates in the elimination clearance. And we estimated an important between substrate variability and between occasion variability in clearance as expected, and the same way as other reported models. And the interesting point that I want to touch on is that we thought that this model will have a better performance on the retrospective data, on predicting the retrospective data, which was collected in the same place, in the same setting, by I would say, the same healthcare providers. So, but interestingly, the model, our model, did not perform better than the other models in the first occasion. So, really, after the third occasion, the model achieved the best performance. Well, I won't say the best. It achieved a good performance. So it definitely it was we have different ideas to explain this. I will not get into detail, but I will say that the variability in the population is definitely important, and even a model developed in the same population does not ensure that it will be like the best possible model to apply in the same setting moving on to the future. So in a context of some questions that arise in a context of high interindividual and interlocation variability, how much data we need to develop and validate the model for MIPD, and to trust on the projections and to optimize those. Is there a reasonable sample size to adequately represent the target population? So I think here yes. Thank you, Amparo. So some things that are bringing to solve this are definitely what Ron touched on, continuous learning, and the other is multimodal approaches. And I will not get into detail, but because it was already touched on by previous speakers. But so I think by applying multimodal approaches, we provide more flexibility into the model. We allow more flexibility into the individual parameter estimations. We allow different distributions to be chosen by the model depending on the individual data. And therefore we have more flexibility. Of course, this is not a non-parametric approach, but at the moment we integrate the data, we have more support points, and we are less constrained by the distribution of one single model. So this is just to show, and this is a work we did in collaboration with Sebastian Wicha. And so we choose the 5 models with worst performance. We did a lot of evaluations, but only to show you, by choosing the 5 models with worst performance and implementing MSA and MAA, so model selection and model averaging, they had already a very good relative bias compared to the individual performance. So this is only a point to touch on that. These methods can definitely improve how we handle and how we implement more informed precision dosing. Definitely, external evaluation is something important, and we have worked on developing tools to automatize this evaluation, because it is definitely time-consuming. If you think about integrating data occasion by occasion for more than 20 or 30 models, and to get estimates of precision and accuracy at each occasion, and then make simulations, it is very computer intensive. So we are developing a package that will be soon available that performs this estimation in 3 steps, in 3 lines of code. And we did this with Ignacio Alvarez Castro and Nicolas Schmidt, which are a scientist and a political, a statistician and a political scientist, respectively. And the last point is platforms. Definitely, it's important to think about the platform on which you will bring your model into the clinical practice. This is a very nice paper I recommend reading by Chloe et al. How this platform can integrate the data and provide the model to let the model to allow the clinicians to actually use the models. And there are different tools, of course, available. We thought that it would be nice to have our own tool because it provides us with the flexibility to implement any model that we want, that we can be working on, that we develop into a platform that allows to implement this Bayesian forecasting approach in the hospital. So we work together with Laura Gonzalez and a group of students of computer engineering to develop a prototype to implement more informed precision dosing at the university hospital. This works as so we mainly work as a front end that is intuitive and friendly for the user, and this uses Monolix and the Monolix suite at the back end to perform the Bayesian forecasting and provides providing the models for any drug that we are interested in working with. So the model actually creates integrates the data and the estimating individual parameters, allows to perform simulations and reporting. Yeah, that's something to show the model. The platform is a web-based platform, and we are doing the first. As a prototype, we are working with the nephrology center to evaluate this in the clinical practice. That we are integrating the tacrolimus model and also with the multimodal approaches to evaluate the performance of this tool and to evaluate the use by physicians to decide to take the decision on the first dose. So to wrap up, I would say in the interdisciplinarity is essential for MIPD success. There are several key challenges to be solved on site, so it's definitely important to have a critical mass that is aware on more informed precision dosing at the clinical site. And yeah, we still have a lot of work to do to prove to the medical staff and the healthcare providers the benefits of MIPD. And with that, I want to thank you for your attention. I want to thank the university and the sponsors of the different studies, and thank to Simulations Plus to organize this webinar, and my colleagues, speakers and chairs. Thank you.
Thank you, Manuel. I'll ask everybody to turn on their cameras while Jasmine sends out the next poll question. Thank you, everybody, for your fantastic presentations. Jasmine?
Sure. Our last poll has 2 more questions, and I'll give everybody a couple seconds to answer those. All right, I see answers coming in. And I'll go ahead and end it there. Go ahead, Amparo.
Anna, do you want to start with the questions that we have in the chat?
Yeah, I can take one of those. So the question is, nice presentation and talks. I have a question. What does the term precision means in MIPD term? Precision is usually the scatter around the prediction. Where is the accuracy as the measure of prediction bias then in the whole story with precision dosing? Thank you. Who would like to take on this question?
Maybe
right,
I
Sebastian, you
start
can
and
go.
others can chime in. Yeah, it's of course correct. I mean, it should probably say model-informed, accurate, and precise dosing, which probably sounds a bit clunky. And then I think precision was kept and accuracy was stopped. There are other terms. I think MIPD is the one that now has emerged a little bit. There were others, Bayesian forecasting, model assisted TDM or something like that. It's terminology, and terminology evolves, and maybe MIPD will also evolve further. So the question is correctly phrased, but yeah, it's a term. Yeah.
I will add
Somebody
that.
would like to add something?
Yeah, I will add that I think we or the community have chosen model-informed precision dosing to emphasize and to make it similar to precision medicine, which is a term that is already very established in the medical community. And I think we can see model-informed precision dosing as a cornerstone of personalized and precision medicine, if you think about the importance of treatments, of drug treatments, yeah, in the clinical practice.
I guess I can also add that Anna and I are involved in a project that's looking at the terminology that's used in pharmacometrics, and she's smiling because this is one of the I guess terms where there was really quite significant debate amongst the working group, and I would just flag to watch this space. I think, as Sebastian hinted at, there will be the possibility that this term will evolve, and as part of this project that Anna and I are involved in, really around I guess, the use of precision and accuracy also in other fields outside of pharmacometrics, and just ensuring that we try and limit misunderstanding of these terms as much as possible. So watch this space. And I don't know, Anna, if you want to add more to this.
I have no opinion today, so I will I will defer to today's speakers for that. Thank you, Sophie.
Very good. Do you want to?
Should we move to one more question before we finish, Amparo, or you want to close it because we are already 1 minute?
I know. Let's do one more question. And I think Manuel is typing the one that was sent in the chat, so that should be available for all.
Okay, so another question would be, when we use the term of precision dosing that could be considered off-label use, what will be the implications of that? Ron, go for it.
I can try. I mean, for a lot of drugs, it might not necessarily be off-label. There might be already in a label that says, okay, we recommend this dose, but using TDM, you can alter the dose, perhaps with a maximum dose or something. It depends on the drug. So it might not necessarily be off-label to actually do this. For some, it might be, but in many of those cases, it's already happening anyhow, where a doctor based on TDM is already changing the dose. So this is, I don't think the fact that you're doing it using an algorithm changes that much to the current practice. I think you're doing it just more, I want to say precise, but accurate.
We're afraid of using the term until we get the new definitions out, right?
Yeah, now I'm getting scared.
All right. So, Jasmine, would you like to wrap us up?
Sure. I want to thank all our speakers. For any questions we weren't able to answer, our BD team will reach out following the webinar. And we're also going to be hosting 2 more webinars on QSP and oncology, and then DDIs. If you want, you can go register for them on our website. I put a couple links in the chat. You can also follow us on social media, and wanted to let you know that the webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.
Thank you.
Thank you.
Thank you.